Andhra Pradesh, and I completed my MD from Gandhi Medical College and completed my DM from Nizam's Institute of Medical Sciences. So where we were doing various phases of clinical trials. When Balaji asked me, I thought that 20 years back exactly I did one phase 1 study. I was a part of phase 1 study, not I did it. I was a part of phase 1 study. And then I went and looked at what happened to that molecule. Luckily in 2019, it has come into the market. It is an Indian antibiotic. So I take few examples of that. I'll show my experience, how we did it, and I'll walk through the all phases of clinical trials. And you can interrupt me at any time. Since I'm a teacher, basically any interruption, I won't mind it.
Thank you, sir. So you can switch on to the presenter mode and you can go live. Yep.
When Balaji asked me to give a presentation on clinical evaluation of drugs across phases of clinical trials, then I thought that normally whenever I used to go to these type of lectures, there will be a lot of dumping of information that these are the phase 1 trials, that this and all those things. But somehow I was not comfortable at the time. And luckily, yeah, because I worked there, I got an experience. But I'll be telling about few of my experiences, but not the exact data what I have done it. So this is my disclaimer that I only tell my experiences.
Sir, can you go to presenter mode?
Is it visible?
No, you have to click on the presenter mode in your PowerPoint. Down at the bottom right of the screen.
Oh, this one.
Yes. Thank you. Yes. Thank you.
Okay. So normally the clinical trials are the research studies that test the new medications, medical treatments in people. Actually, they are the final step in the R&D process for evaluating new treatments. They are the definitive way to determine whether the new treatment is safe and effective. Clinical trials also help to improve the quality of existing treatments. Anyone who meets the eligibility criteria can participate in clinical trial. Normally, clinical trials are conducted in 4 phases: phase 1, phase 2, phase 3, and phase 4. And there are so many explanations for the phase 1, phase 2, phase 3, and phase 4. And there are so many synonyms are also there. Now, basically, what is a phase 1 study? Phase 1 study is a first-in-man study. Once the molecule underwent extensive preclinical testing, and if they found that the molecule is a candidate, is a lead molecule, and then they will do a clinical trial first-in-man study, that first in human being. That human being can be a healthy volunteer or can be a patient. It all depends on the type of molecule. So phase 2 study is a first in patient study. Third phase is called a therapeutic trial or a registration trial. And the phase 4 is a post-marketing study. So in the phase 1 study, we do safety studies. Mainly it is concerned of safety. Second phase is exploratory efficacy studies. Third, phase 3 is confirmatory efficacy studies, and phase 4 is again a safety study. So there are 2 safety studies and 2 efficacy studies in the phase 1 to phase 4. There are the synonyms of these clinical trial phases. Phase 1 studies are safety studies. They are also called pharmacokinetic studies. They are toxicity studies, tolerability studies. When it comes to the dose, it is a dose escalation study and a dose ranging study. So we'll talk about this in the coming slides. The phase 2 study is a pilot study, pilot efficacy study, exploratory efficacy study, dose finding studies, and sometimes it is a dose titration studies. And phase 3 studies are the pivotal studies. They are the confirmatory efficacy studies. They are registrational studies. They are the fixed dose studies. So each phase has got so many names because there are so many endpoints will be there in each and every phase of a clinical trial. When it comes to the Phase 4 study, it is a post-marketing study. When it comes to the dose, it is a study based on by seeing the adverse effects and the efficacy of labeled and unlabeled dose. It is a pharmacovigilance study. It is a market seeding study. We'll come across in the coming slides. When you pass through the slides, we'll talk about all these terminologies. And when it comes to my experience, phase 1 is a step study, the most important step in the drug development, especially in the R&D when it comes to the clinical trials. It is broadly, it is a safety, tolerability, efficacy, and pharmacokinetics. So immediately after testing from the animals, then when we move to the phase 1 study, we will always be concerned with the safety of the molecule. Then we have to see the maximum tolerated dose by any human being. So that is the dose where we cannot use beyond that we cannot use the drug. Third one is if the drug sometimes shows an efficacy, say for example, an anti-diabetic drug or an anti-hypertensive drug. So it will have some efficacy. So we can easily capture that efficacy even in the early trials also. And finally, the most important thing is the pharmacokinetics. Because basically the drug development, everything goes by pharmacokinetics. Because 30% of the molecules, they fail because of lack of showing, not because of efficacy, not because of showing favorable pharmacokinetics. Phase 2 study is again the same thing which is being done in patients. This is an early study, early patient study, where they will be involving a very few numbers of patients, roughly around 100. And again, these phase 1 studies are divided into 1 or 2 stages, they will not do everything in one stage. Even the phase 1 study they will not do everything in one stage. They will do stage by stage, stage by stage, stage by stage. And again, phase 3 study is a fixed dose study comparing with the available product in the market. And phase 4 study is again is post-marketing study. Once after phase 3 study, they will go for a registration in the India or USA or Europe. Once it gets a regulatory approval from the respective authoritative in that particular country, then after that, then they will go to the phase 4 post-marketing study. It is mainly based on the safety. So again, going back to this, in the phase 1 study, it is a safety study. Here we can construct doses from minimal dose to the maximal dose. That is a dose construction study. It can also be called as dose construction study. And it is also called as dose ranging study. And it is also called as dose escalation study, where we escalate the dose from the lowest dose to the highest dose. That is a maximum tolerated dose. Here in the dose finding study and the dose finding studies, what is the appropriate, what is the normal doses that we can predict that can be taken in the patients based on the phase 1 study. Because it also involves a little bit of initial efficacy. Efficacy cannot be measured if they are not measured in patients. That same efficacy can be measured in case of biomarkers, the markers that from which we can assess the initial efficacy. And once we fix 3 or 4 doses from the dose range, and this begins a dose titration studies. We have to see the titration. Whether we have to go for an optional titration or forced titration. And again, we have to see the maximum tolerated dose in the patients. The phase 1 study is nothing but, say for example, if I want to do a blood urea or a serum creatinine in the lab, what I have to do? I have to first do a developmental study. So I have to go by the lowest level that I can detect a serum creatinine to the highest level that I can do a serum creatinine. Similarly, we do a similar type of study that Anybody want to ask question?
No, sir. There is nothing in the chat. You can go ahead.
Okay. So it is a dose escalation study or dose ranging studies are similar type of things where we construct the doses from the lowest dose to the highest dose. And since I have a little bit of experience in the development of this levonorefloxacin, roughly around 2003, 2004, when I was doing my MD, I was a medical officer. So I used to do the night duties in Nizam Center of Medical Sciences when these trials are there. So I got an opportunity to participate in this trial. And when Balaji asked me and when I looked into back, because I want to tell that phase 1, but luckily this molecule has come into the market. So I'll talk about my experience about it, not the exact company core data sheet, which I don't have it, which anybody will not have access to it. Company core data sheet will have all the information that what I don't know also. Levonorefloxacin is a quinolone type of antibiotic. Levonorefloxacin is IV and it's prodrug because the oral form of levonorefloxacin is not reaching a similar concentration when given it in the same form. So a prodrug has been prepared. A prodrug it goes into the systemic circulation, then it undergoes metabolism, then it becomes a levonorefloxacin. It is having a broad spectrum against both gram positive, gram negative, atypical organisms, and anaerobic pathogens. The second statement tells that oral levonorefloxacin oral drug has got 90% bioavailability similar to intravenous. Say for example, if the dose of IV drug is 800 mg, then approximately the oral drug will be around 1000 mg. And they have done a hepatic impairment study, and they have demonstrated it. The therapeutic doses need not be altered. The phase 2, since the phase 2 and phase 3 trials are successful, and now it is marketed in India, it became the treatment of option for multi-drug resistant gram-positive organisms. And coming to the phase 1 study, this is a safety study, tolerability study, efficacy study, and a study which is capturing the pharmacokinetics. This study was conducted in healthy volunteers and in patients. So the starting dose of this phase 1 study is typically much lower than the dose that was effective in animal studies. Very, very lower. The dose is started at a very low and gradually increased to find the maximum tolerated dose, the highest dose that can be given safely to humans. The volunteers during the study, the volunteers are closely monitored for the adverse effects. And the data on the step safety, tolerability, efficacy, pharmacokinetics is collected throughout the trial. And a few references, few clinical trials, I like phase 2, extended phase 1 studies. All this I have taken it from this article. A phase 1 usually is an open label study. It can be double blind also. It all depends on the formulation. It is a single ascending dose study and a multiple ascending dose study to evaluate the pharmacokinetics of this particular drug administered intravenous oral in healthy male adult volunteers. And in this study, they have done the phototoxicity assessments also. The reason for doing a phototoxicity assessment is these fluoroquinolones are known to cause phototoxicity during the early doses. That is the reason, and phototoxicity causes skin blisters. So the reason for doing a phototoxicity study is to evaluate whether it causing any phototoxicity. If it is causing phototoxicity, then the patient will be advised to wear full shirts and use some SPF creams of 30% or 50%, depending on the radiation of this light. In one of the information that was available, 12 healthy male adult volunteers received intravenous dose of levonorepinephrine at a dose of 250, 500, and 1000. How did they arrive at 250, 500, and 1000? Is based on the laboratory tests that have conducted against the bacteria, where we can see that the inhibitory coefficient of 50% or inhibitory concentration of 90% is usually taken as a benchmark for assessing whether. And how did they arrive at 250 will give a drug level that is above IC50 and IC90, or 500 above IC50, IC90, or 1000 mg above IC50 and IC90. Since this is a concentration dependent drug, it exhibits a concentration dependent pharmacodynamic effects. So it should be more than the IC50 should be more. It should be above the IC50 and IC90. And that is the reason why. But how did they arrive at 250? How did they arrive at 250? The answer is based on the animal studies. They know they have calculated the human equivalent dose from the animal studies, and they will not directly go from the directly go from the dose. They go much lower. Since these are the very few studies done in India around 20 years back. We had an opinion that we should not rush the doses. We should not rush to a higher threshold level. So we have to be on the safe side, and we have taken a lower dose, starting dose, and then we escalated to a higher dose. And in the multiple ascending dose studies, they have taken 2 doses. They have given it multiple times for 10 days, once daily for 10 days, and they have seen the pharmacokinetics and safety there. What are the safety analysis usually we see? We see the physical examination, vital signs, cardiotoxicity, hematological toxicity, biochemistry, liver toxicity, renal toxicity, and urine analysis, because it gives a complementary to the above tests. And in addition to that, radiography to see whether any imaging techniques to see the CNS functions, CNS toxicity has occurred or pulmonary toxicity has occurred. Another important thing after safety, the another important thing that what we calculated, what we observe is the pharmacokinetic parameters. The pharmacokinetic parameters are usually non-compartmental, because we don't assume, because we go by null hypothesis, and we will not assume any compartment like 1 compartment, 2 compartment. And all this compartmentalization comes after the phase 1 study, not before the phase 1 study. So we go by the non-compartmental pharmacokinetics, because that is the null hypothesis. So we measure the maximum concentration, time to reach the maximum concentration, total bioavailability during the 24 hours of time, that is AUC, what is the half-life, what is the mean residence time, what is the clearance, and what is the distribution, and what is the metabolite. Everything is known from the phase 1 study. What is the major metabolite? Because they have some idea, because they were developing the drug, they have the idea. Then they will find out how the liver, how the human liver behaves, and it gives a metabolite. And in the multi-dose study, since 10 doses are given, there will be 10 peaks and 10 troughs. So they will measure the trough to peak ratio. Trough to peak ratio, in addition to the half-life, clearance, volume of distribution during the 10 dose study or 10-day study. And this study was conducted at Nizam's Institute of Medical Sciences by Dr. M.B.R. Naidu, who is the principal investigator, and he was the head of the department of Nizam's Institute of Medical Sciences. Later, he served as dean of Nizam's Institute of Medical Sciences. Now he is superannuated, and he is relaxing at his house. In addition to that, he actively participates in the IMPA group and he gives lectures to the PG students. in the study that what we have done, it was a prospective, randomized, double-blind, placebo-controlled, parallel group study. You may ask, why a placebo-controlled, why a double-blind? Why can't you directly go with the drug? Basically, the placebo control is serves as an internal validity. Whatever the procedures that we are doing with the drug, whether the same procedures have been followed with the placebo or not. So if there is any discrepancy is there, it will be shown when there is a comparison between the placebo and the drug. And here, as I said, that we evaluated the safety, tolerability, pharmacokinetic profile of multiple dose study because it was given for twice daily for 5 days. However, the doses were different because I told you the company company studies a wide range of doses, and everything will be documented in the company core data sheet. So here we evaluated 500 mg in 6 patients, and we used 3 placebos. 600 mg in 6 volunteers and 600 mg in 8 volunteers for a 5-day study. So each group, when I select a 500 mg group, I'll select 3 volunteers and 2 will be given the active drug and 1 will be given. So one will serve as a control and he will be given as a placebo. Because sometimes it is possible to do a blinding study, sometimes it is not possible to do an unblinding study and initially whenever a sponsor develops a new drug, usually he goes with an IV formulation because making a few number of tablets is very, very expensive, time consuming. It requires a lot of manpower also. That is the reason why they go with an IV formulation. Once the IV formulation establishes the safety, then automatically say based on the indication, what is the proposed indication? Say if it is a diabetes, hypertension, actually we have to develop an oral drug. Otherwise, the drug will not, patients will not accept the drug. Since this is an antibiotic, usually antibiotics are given for 5 to 7 days or 10 days or up to 14 days. That is why they are doing a multi-dosing study because they want to see the steady state pharmacokinetics. Say for example, so it is divided into 1-day study and 5-day study. So volunteers will be checked in for 24 hours prior to the dose, and 48 hours of the last dose they will be checked out. They will be discharged from the unit, clinical pharmacology unit, and every morning till 7th day they will be after the last administration they have to come to the follow-up, and we used to examine the patients. So totally a 1-day study would require a 4 days hospital stay, and a 5-day study would require around 8 days of hospital stay. And clinical pharmacology unit is nothing but a hospital in hospital, a mini hospital in hospital. So this is the study preparation. This is the crash cart. It is nothing but an ICU. In simple terms, if you want to tell that the clinical pharmacology unit is nothing but an ICU unit. So it has a crash cart, how the volunteers have been administered the drug. So many, since we were doing it for the first time, we are also anxious. So many, we used to be there throughout the 24 hours for 8 days. This is how the vitals are monitored. The study drug is being prepared for infusion. This is how the infusion of the new drug, how the drug is being infused. They are monitoring the patient safety. We are recording the ECG. We recorded the Holter monitoring 24 hours. And study is monitored by a consultant, who is a physician and an anesthetist. And myself recording the data. Blood sample collection process. See, basically, we'll talk about the steady state pharmacokinetics. The steady state pharmacokinetics, these are the 10 peaks that we have observed. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And after that, after the 10th dose, we have followed. So this is very, very important, because steady state pharmacokinetics is nothing but the amount of drug that is being infused, and that is equal to the amount of drug that is being excreted from the eliminated from the body. So after the 10th dose, what happened? So it remained, we measured up to 48 hours. So that is the period when we discharge the volunteer. The phase 1 safety overview is there were no SAEs, serious adverse events, no hypersensitivity reactions, no idiosyncratic reactions, no clinically significant specific ADRs. These are called AESIs. AEs of special interest. No dose-related ADRs, no specific trend in ADRs, and we finally concluded it that this drug can enter into the phase 2. Going back to little. So this, once the 500 studies safety results are, unless and until we don't get a 500 mg safety result, we will not do a 600 mg safety. Once the 600 mg safety results are not collected, we will not proceed to the further study. So this is a cohort studies. So each cohort, we have to get the full information about the safety of the volunteer. Then we have to proceed to the next dose. And not in our study, somewhere else they have conducted a phototoxicity study also. And these volunteers were, after giving a multiple dose after participating in a multiple dose study, they will be, their forearm or something, the forearm will be kept under a light simulator or an ultraviolet bulb, which is having both A and B, or this solar simulator, and they will monitor whether any reactions by using a visual analog scale or a digital imaging. The results of the photo testing are typically expressed as percentage increase in the erythema from the baseline. It means the patient will undergo a baseline study of phototoxicity also. Since the drug is known to increase the QT, so cardiac safety has to be established, and they have done a special study, a double blind study, a double dummy study. Double blind, it's a placebo controlled. It has got an active drug. It has got a levonordic fluoxetine. So one placebo for moxifloxacin, another placebo for levonordic fluoxetine. That is why it is a double dummy. And it's not only a double blind, it is a triple blind study. Basically, the investigator is blinded, the patient is blinded, and the assessor who is assessing the QT interval is also blinded. And here they have used supratherapeutic doses. And they came to know that some of the phase 2 studies that they were planning to give 1000 mg BID. So automatically it comes to 2000 mg. So they have seen up to 3000 mg in a triple blind fashion. First, they have constructed the safety of 1800 mg, 2200 mg, 2600 mg, and 3000 mg, 3000 mg. In again 2 plus 1, 2 plus 1. Sorry, 3 plus 1. Here it is three volunteers for this particular dose and one volunteer for this particular dose. So once the study results of 1800 are is not collected and whether safety is not established, they will not go to the next dose. So it will take a long time to do a phase 1 study. And finally, they have selected 2600 dose and they have conducted a study with 400 mg, comparing with 400 mg and placebo in a 13-day study. Day one, like it is a crossover study, and they have done it in three runs. First run is they have given it in levonorepinephrine. Second run is in with the 400 mg moxifloxacin, and third one is in placebo. I'm not talking about the results of this study, because our main idea is in the conduct of the clinical trial. And they have done one important study in healthy adult human subjects. They have compared 1000 mg and 1000 mg twice for 5 days, and 1 dose study and 10 dose study. And they have collected the intrapulmonary liquid. That is intrapulmonary liquid. We need an anesthetist to do a bronchoalveolar lavage and then collect the fluid, and then we have to measure the concentration of the drug. One of the reason is probably they want to establish that this drug can be useful in case of community acquired pneumonia or some ventilator acquired pneumonia in the future case. Finally, after doing all the studies, and they planned that, so we'll go to a phase 2 study. We have got the sufficient data regarding the IC50s, the doses, the safety of the doses. See how plannedly they have done it. So in the phase 2 study, they have selected an optimum dose of 1000 mg 2 times a day. Normally it would be a oral dose. So they have selected a condition called acute bacterial skin and skin structure infections, and they want to test this drug in that infections in a double-blind manner at the placebo control. Here you may ask, why a placebo control when there is an infection? Because skin and skin structure infections are known to have a self-healing capacity is up to 50%. That is the reason, one of the reasons why they have gone for this. And placebo, I already told you that placebo increases the internal validity, and comparing with the drug with an active comparator will increase the external validity. Here they have measured the clinical cure, microbiological cure, AEs, SAEs, AEs of AESA, AE adverse events of special interest like QT prolongation, hepatotoxicity, nephrotoxicity. And once these study results are clear and they are confident, then they went to the phase 3. And here they have compared with a active comparator, linezolid. So comparing with the linezolid will increase the external validity of the particular trial. And Voca to become the first Indian company to get the nod novel antibiotic. And what is their future plan? I don't know exactly, but it's only my prediction. Since the fluoroquinolones are known to cause the tendon ruptures and peripheral neuropathies and psychiatric events, probably they may conduct a prospective cohort studies, like patient support programs, like which doesn't usually happen in India, but they do usually happen in Europe and America and Canada. And some other post-marketing studies, some other post-marketing clinical trials, they may do it, and they may get huge number of data like 1 lakh data, 1 lakh patients data. It takes a long time. So what they will achieve from this study? They will achieve the safety, and they will get a new indication for this existing drug. That is one of the importance of pharmacovigilance. With this, I thank Balaji for giving the opportunity, and I thank the audience for showing their interest in attending my lecture.
Thank you, sir. So now we'll open up some questions from audience. So people interested in having some questions can directly shoot the questions. So maybe I can ask a question, sir.
Sure, sure.
So when you told about this phase 4 studies, which they might plan.
They might plan.
Yeah. So what do you think the status of these active surveillance studies in India currently at this moment? Let's have two divisions. One is the pharma products, the other one is vaccines. So where do we stand at this moment? Other than the passive surveillance, which usually happens as a part of spontaneous reporting in PV,
Yes.
these
Yes.
active surveillance programs, where we stand now currently?
Currently, we are not that in that pace. Very few studies might be occurring, but every sponsor is probably may not be doing the same thing. Yeah, they are doing in a small scale, not in a big scale. Like patient support programs where they have to do it for, like in case of large molecules like immunosuppressants and biologicals. They will do it for a long time, and they will also gain some benefit of that, because a new indication for existing, they'll find out a new indication for existing.
Do you think that the innovator companies which are doing trials in India, the big giants, because there are very few Indian companies which are doing innovative trials, they are mostly into generics, we all know. But so now the scope is coming up, and even the Indian companies are doing some innovative trials. So what do you think about these innovative companies? Let's take, for example, Pfizer. So do they do PMS studies in India to follow up actively?
No, because I worked in quintiles for 2 years. I have not seen an Indian study.
So do you think?
Yes, a study that is being conducted in India. Sorry, not an Indian study.
So do you think that the regulatory scope needs to look into this aspect to make it compulsory for innovator drugs that are marketed in India to have an active surveillance program running? You think so?
Yes, that is the need of the hour. But since many innovative products are not being coming, we hardly see a innovative products from India. That recently we had this molecule. I have seen because my knowledge may be limited. I have seen this molecule and COVID vaccine.
Oh, absolutely. Only I have also seen only these two molecules, because there was one malarial program that was going on with Dr. Reddy's, but I'm not sure how far that has come true to be an innovator product.
Yeah.
But so why do you think we are not able to produce innovative drugs? Being a giant in generics, why do you think we are not able to do it?
So maybe active research, promotion from the government.
Or do you think the clinical pharmacology setup in many of the good government colleges? See, for example, let's not take the central institutes. They do have a good phase 1 unit. But what about the other leading government colleges? They don't have a phase 1 unit to get an innovator trial that can be done. So do you think that the need of the hour is that every government college should have such setups?
So it all depends on the number of innovators that come that approach. Basically, if a medical college or something like institute is not doing, they are directly going to the CROs. The CRO setup, their CRO setup is there. They will hire a clinical pharmacologist from a medical college.
Correct. So do you think then
So-
the curriculum needs to be a little bit tweaked between an MD and a DM pharmacologist?
Yeah, time will tell. So if I have not joined DM clinical pharmacology, probably I may not worked in that, and
Yeah,
I have not
because
appreciated since
yeah.
it is a 20-year time for me.
No, actually, even now it is true. Only DM pharmacologist gets to know about these clinical trials in an active way, in a real world situation with when he's associated with the college. But I don't find an MD pharmacologist or a professor or an academician who is still an MD pharmacologist doing clinical trials in a top-notch way. That's not happening in the country.
Yeah, few people are there.
Yeah, few people are there. Primary, tertiary centers do it,
Few
but
people are there.
yeah.
So
So
before that, if we are not into clinical trials, then what I have been doing is we are developing few instruments. So indigenously developed instruments. So validation of that instruments. So that is another way where we can do that. If trials are not coming, what we have to do? We have to write some protocols. We have to do validation studies of biomedical instruments.
biomedical
So
instruments. Yeah, that
so
can be
we
a
have
takeaway message. Yeah.
yeah, because Nizams is famous for validating biomedical instruments, and I was a part of that for 4 or 5 biomedical instruments.
This is a good takeaway. So if you don't get to involve directly in a trial, you can start working on the other aspects which can aid in a clinical trial.
Yes, yes. Basically, that one we developed one. Actually, we were quantifying the vasoconstriction. It was for a biological molecule, for a nitric oxide generic.
Okay.
So we developed that instruments. We are part of the development program along with the biomedical engineers, and we did a bioequivalence study with the nitric oxide molecule.
Okay.
So currently we are developing a pupillometer, binocular pupillometer. Normally, the commercially available pupillometer is it measures the interpupillar distance only. So we want to find out the real-time pupil size, so that we can do a bioequivalence study with some mydriatics or myiatics coming.
Thank you, sir. Thank you. So from audience point, any questions? If not, we'll wrap up the session.
I think there was one question posted in YouTube. Can you check it out?
Yes,
On
yes, sure. Let me just check it out. Oh, in YouTube. Okay. Okay. So namaste, Dr. Ramavan, sir. What does double dummy means?
Yeah. So when it's a 3-arm trial, one arm is placebo, one arm is the active drug, and one arm is the comparator. One is the test drug, and one is the reference drug. So I want to do a double-blind study. So there will be unequal number of tablets. Sometimes there can happen an unequal number of tablets. So that if we don't use a double dummy technique, the blinding will be easily broken, because if 2 patients start, one patient say I'm getting 4 tablets and you're getting 3 tablets, automatically the blinding will be broken easily. To maintain the blinding, we use another placebo in that particular group to maintain the level, to level the number of tablets in that particular trial. By this way, we can preserve the blinding.
Thank you, sir. So one more last question from my end. Do you think placebo is required?
Yeah, placebo is very much required, because placebo is the internal validator.
Okay.
Even if you do a chromatography, you will use a known compound, so that whatever the extraction of that known compound will also be influenced by the process. If the process is not correct, and if you are extracting a new chemical, how do you know that your process is foolproof?
Okay.
So for that, you add a known concentration of particular compound, and you see how much it is being extracted. This is similar to what we do in clinical trials. So I do a ECG study. In that particular study, if my ECG studies are not good, The ECG machine is not good. Whatever the results that coming with the tested drug will also come with the placebo. So that will increase the internal validity. Say for example, I have done a 8-day study. 8-day study means the volunteer will be given a controlled food. All the volunteers will be in one big hall. They will be in ACs. Of course, they will be seeing TVs, they will be playing cards, they will be singing, something like that. But they all have a controlled food.
Typical
So,
BAB center setup. Yeah.
Typical BAB center setup. So what will happen? Some food poisoning has happened. Or the food that the particular day the food is not good. So there was some infection to one person, it has spread to other persons. How would I know? Unless and until if I have a placebo on that particular day, so okay. Then we'll find out. One day that one volunteer was watching Australia, South Africa match where it was a huge match for around 430 runs to chase, something like that.
And South Africa changed it and won it. Yeah.
We are watching that match. One volunteer got tachycardia. And we have to know why he got tachycardia, because it is anxiety.
Correct.
So after that particular, we have stopped doing matches to the volunteers. neither we were showing horror movies also. We were showing only love movies.
So this might hold good for many drugs, but what about oncology trials? Do you think placebo would be no?
Oncology trials definitely need a placebo.
Is it?
Because one of my senior oncologists, what he tells is, if you don't use a placebo, then you are thinking that your drug is working. Don't think like that. You have to go by null hypothesis.
But if you do a phase 1 in a patient population itself, do you still go with the placebo?
Yes, still go with the placebo, because I told you, they will be housed.
Okay.
They will be served food. Say for example, one patient has got diarrhea, or one patient has got vomiting. So the same thing happens in placebo. See, usually you see that in most of the medical literatures, drug promotional literature, you see drug X is superior to drug Y, but it is having a placebo-like side effects.
Yeah.
It suggests that it is a 3-arm study. Drug X is superior to drug Y, but whatever the adverse events the incidence of adverse events that occurred with this particular drug is similar to that of placebo. So it shows how tricky they have written it.
Yes, sir. Thank you very much, sir, for that, because many people think placebo is not required. So the reason I asked that question is, what's the importance of placebo? If you coming from a legend like you, that placebo is of most importance, people will understand what's the real importance of placebo. Yeah. So I think we are good here. So, sir, thank you very much for your time. It was a great session, because in industry, people work in different trials, but they don't get to see this basic stuff which they really need to understand. Because I have seen people from scientific writing, from pharmacovigilance, from phase 3 trials, phase 2 trials. Still, they are not aware of how a phase 1 trial is being done, to be very frank. Some things like SAD studies, MAD studies. So these are all good sessions where people can now go back, use this as a placeholder, and they can read more on phase 1 trials, how to develop a new product. So it will help everybody to understand drug discovery in a better way. So it was a great session, sir. Thank you for your time. So on behalf of DTRAS, I like to thank you for your time, and I just asked you, sir, can you do it? And you accepted it. It's a pleasure for me. Yeah.
Basically, actually, I would have done it different way, but you have already given the title, so I have to justify the title.
Yeah.
So I was about to present only phase 1, but not phase 2 and phase 3. I would have not been done doing this across the clinical trials. But since you already given the title, then I started working. Initially, I did not get this idea for first 10 days. Last 3 days, I will be working.
Okay. So we would like to welcome you again, sir. We would love to see you in the forum, maybe sometime later with some other new topic, which will help everybody in the pharmacology industry.
Yeah, and thank you very much.
Thank you very much, sir. Yeah. Thank you. Thank you, everybody.
Thank you, everyone.